Hello, my name is Arya, and I, alongside with Matthew, Maria, and Anna, will be talking about cancer and the environment, chemicals and pesticides in food in PCB 4234, Biology of Cancer, here at Florida International University during the spring 2024 semester. This topic will explore in detail how the environment around us humans influences how we may contract cancer, and if there are any specific types of cancer that will have a higher chance of contraction because of such factors. In addition, how different types of chemicals and particles that are present in foods will have a both negative and positive effect on cancer. Introduction to cancers. The rule of thumb. It is vastly agreed that cancers are defined as an abnormal cell growth. While there are many different types of cancers, each has their own risk level, possible causes, and the treatment that could be used. Breast and lung cancers are among the most common types of cancers that are out there. Cancer and its factors. There are both man-made and natural ways a person may contract cancer. Radiation from UV rays from our sun and exposure to toxic chemicals such as benzene, arsenic, and vinyl chloride are such examples. Most exposures can be avoided by having clean and healthy lifestyles that promotes diet and well-being of you and others. Environmental factors can increase the risks of cancer, which will be moreover touched on later. Ecology of cancer. Ecology is interpreted as the study of the organisms and the environment around them that they get to interact with. Cancer involves both an ecological and an evolutionary process. The interactions between the natural environment and cancer tumor cells are such processes. Within a tumor, hormones are used to signal cancer cell molecules. Since cancers are complex ecosystems, malignant cells may coexist alongside each other and work together in unison within the microenvironment with the host cells. Cancer through an ecological lens. The way cancers work can be better identified by the help of ecology. We can understand its operations better. Hypoxic microenvironment are strong among cancer cells, hence why there are a lot of competition for cancers finding necessities at scarce locations. Because of this, cancer cells search for new places when competition is lacking and there are enough resources to use. Specific phenotypic features are converges by cancers to look for a higher chance of fitness within the body's ecology. There are parameters for both proliferation and pro-survival phenotypes. Since cancers may be found as living organisms that are interactive with both cellular and non-cellular components of a host's inner systems. Some examples may include the host's nervous, immune, and endocrine systems that are mainly in use for a self-sustainable ecosystem of tumors. Influences of the environment on cancer development. Many possible ways are present for humans to get cancer through many different types of ways. For one, exposure to toxic chemicals that are in specific fields of their agricultural and any other industries such as pesticides, benzene, and tobacco. These can create a way for uncontrollable cell growth to initiate within the body and create tumors. High radiation, such as exposure from X-rays, UV rays, and gamma rays, can change a person's DNA that may cause increased risks to cancer. Diseases and parasites, such as HPV and hepatitis B and C, are among the diseases that could create a distribution of abnormal cell functions. Air pollution is also a factor. Air pollution in the form of carbon dioxide and nitrogen dioxide may cause a respiratory illness such as lung cancer. High intake of processed foods and a lack of vegetable and fruits in your daily diet could cause obesity that is associated with a higher rate of cancer. Carcinogenic substances such as formaldehyde and asbestos can involve a higher risk also with developing cancer such as mesothelioma or lung cancer. Cancer and foods. It has been believed that there is a connection between the types of nutrition humans consume and how much of a certain product that they consume with the possibility of contracting specific types of cancers within the possible near future. As we progress through this presentation, our group will touch up on this topic with much more detail and with examples being provided to build up on this topic of interest.
Through this presentation, we will be learning about carcinogens that are commonly found in food and the effects they have on humans. We will be doing this through the use of research and other sources in order to understand the risk and be able to distinguish different sources where these can be found. Carcinogens are considered to be sources that are able to interact with the DNA and cause mutations, which lead to cancer. It is important to understand the role that carcinogens have in food, as many of them are found in food items that are commonly bought and consumed. Preventing the consumption of these items could limit the chance of developing cancer. Some examples of food containing carcinogens are red meat, processed foods, foods containing added sugars, and food cooked at high temperature. In this presentation, we will cover the common carcinogens found in food, how they are formed, and the risk, and how to possibly prevent them. The most common carcinogens found in food are nitrosamines, acrylamides, and polycyclic aromatic hydrocarbons, also known as PAH. Nitrosamines involve processed foods. Acrylamide involves foods that are deep fried or baked at high temperature, and PAH is found in grilled or smoked meats or fish. On the picture to your left, you will see the top 5 cancer-causing foods, with the first being processed meats, then sodas, then fried foods, salmon, and lastly, but not seen in the picture, plastics, like for example, water bottles. While reviewing an article on this topic by Jared McCann on Medical News Today, most of these carcinogens are found in processed meats and can be formed by cooking techniques like deep frying, smoking, and grilling on high heat. Nitrosamines are found specifically on processed meats, like bacon, sausages, and ham, in order to prevent the growth of certain toxins. In a paper written by Young On Park and colleagues, it states that in order to prevent the growth of a toxin named Clostridium botulinum from growing in processed meats, high concentrations of these carcinogens have been found in them. In an article by Adam J. Gushgari and Rolf U. Holden, it was shown that exposure to nitrosamines in food are commonly found in oils, sweets, fat, fish, meat, and vegetables, and can also be found in beers due to the malt being burned by the direct heat. In the literature, it states that ingesting these beers can have increased the rate of pancreatic, liver, and bladder tumors by 32% for males and 22.8% for females. To the right, we see three beers: Old Milwaukee, Olympia, and Budweiser. These three beers are known by the FDA to contain nitrosamine. The most common carcinogen found in food that will be covered in this presentation is acrylamide. This carcinogen is usually found in foods that are deep fried or baked at high temperatures, like cookies, fries, bread, and potato chips. In a research conducted by the National Cancer Institute, it states that exposure to acrylamide can lead to several types of cancer due to it being converted into glycitamide, which is able to damage and mutate DNA. But no clear data has been observed when showing cancer due to exposure from this carcinogen in food. In a research article written by Burhan Basaran and colleagues, report that in a study done in Japan from 1990 to 2013 showed that a total of 2,823 cancer cases were found to have been exposed to acrylamide as foods commonly consumed in Japan, like coffee, green tea, potatoes, vegetable, and cookies. To the right, we can observe a comparison between different breads that have been toasted, with the two most toasted having an X on them, as they are the ones being more exposed to acrylamide due to the high temperatures that they're being toasted at. The last carcinogen covered in this presentation will be polycyclic aromatic hydrocarbons, or also known as PAH. This carcinogen is most commonly found in grilled red meats, as the smoke from the cooking could be highly carcinogenic. PAH is commonly found in foods that are cooked by being grilled, heated, smoked, roasted, or any other techniques that involve cooking the meat or fish at high temperatures. After reading some papers regarding this carcinogen, a specific article from the Agency for Toxic Substances and Disease Registry caught the attention. This article states that the carcinogenity of these carcinogens was studied on lab rats, and it showed a higher incidence of skin, lung, bladder, liver, and stomach cancer. Humans are highly at risk of different diseases when being exposed to PAH. As in an article written by Jenny Rodriguez Sampiano and colleagues, it states that humans are able to absorb PAH easily due to their increased solubility in fat. Based on this article, the reason as to why PAH can be so mutagenic and carcinomic is due to PAH being metabolized by different pathways that are able to bind protein and DNA, causing adducts that lead to biochemical changes. In the picture to the right, we can observe how much The amount of pH concentrated in food increases when the food item is grilled.
Okay, so now that we have a broad picture of three of the most traditional chemicals known to cause cancer in food, it is good to understand what are the immune and metabolic implications of their incorporation to the human body, and just a more detailed biochemical explanation to their carcinogenicity. So first, let's go on to discuss nitrosamines. As it was explained before, nitrosamines are found in highly processed foods, meaning that undergo intense and lengthy chemical processes before being marketed to the public, for the sake of enhancing shelf life, flavor, or both. Nitrosamines are produced by the basic reaction of nitric oxide or a derivative such as nitrite or nitrate, and a secondary amine, as you can see in this picture. When entering the body through the oral route, this compound is metabolized by cytochrome P450, shown in the reaction below, which has a well-known detoxifying role for the organism. And after a series of reactions, as you can see, we end up with this positive charged nitrogen atom linked through a triple bond to another nitrogen atom. This is called an alkyl diazonium ion, and it is the main source of carcinogenicity when speaking about nitrosamines. Now, because of the positive charge that you see here, they will adopt electrophilic behavior and bind to DNA due to its acidic character, promoting protein and DNA alkylation, which, as we know, leads to several mismatches and mutations, and which will later alter the replication of this damaged DNA. This picture right here illustrates how NDMA, one of the most studied nitrosamines found in processed meats, beers, among others as we saw before, is able to cause tissue damage by reacting with adenine and forming 3-methyladenine, causing DNA methylation, which has been linked to silencing of tumor-suppressing mechanisms and promoting of oncogenes. NDMA also has the potential of directly activating RAS oncogenes and moreover, according to a recent in vitro study, inducing apoptosis of leukocytes such as neutrophils through binding to the Fas receptor on the surface of these cells, a receptor which is known to activate programmed cell death. This we can appreciate in this picture right here, which is the mechanism proposed by the researchers who performed the study. Therefore, MDMA and other nitrosamines not only induce carcinogenicity and genotoxicity through direct binding to DNA, but also causes this immunosuppression which contributes to the ideal ecosystem for cancer growth in the presence of nitrosamines incorporated to the body through food. Now we will be moving on to speak about acrylamides. As we mentioned before, there is really no clear data that directly leads to the conclusion that dietary acrylamide causes cancer in humans. However, the International Agency for Research on Cancer has it listed as a probable human carcinogen because of the clear link between its accumulation when cooking grain-rich foods at very high temperatures and DNA damage. Here you can see a molecule of acrylamide which is composed of a carbonyl linked to an amine group. Perhaps we can see a pattern of cancer-inducing compounds derived from amines, as we saw with nitrosamines, and with an alkene group to its opposite side. Now, the presence of acrylamide in foods was actually detected for the first time only about 20 years ago in Sweden, and it is possible that because of that reason, there is still a lot of uncertainty when it comes to the biochemical mechanisms of carcinogenicity that result from ingestion of acrylamide. We do know, however, that acrylamide is formed in foods through an endothermic reaction called the Maillard reaction, which is very popular because it is known to lead to the browning of carbohydrate foods such as toasted bread and French fries. You can appreciate this reaction in this picture. When acrylamide enters the body, it is metabolized by cytochrome P450, just like nitrosamines, and forms this compound called glycitamide, which is an extremely reactive epoxide that will bind to DNA purine bases, causing this DNA sorts of adducts, which are known to be potential starts for cancer cells before abnormal replication would happen. There have also been many studies that have evidence of neurotoxic effects of acrylamides in the central nervous system, triggering nerve cell apoptosis and cytotoxicity through the MAPK to the MAP kinase pathway and the nuclear factor kappa beta pathway, therefore inducing inflammatory response, which, as we know, is an ideal microenvironment for the growth of cancerous tumors and other detrimental situations like oxidative stress. Because of this, it is also linked to the genesis of neurodegenerative diseases. And now, lastly, moving on to polycyclic aromatic hydrocarbons. These are organic compounds that are formed of two or more aromatic rings fused together. They are very well absorbed by the body when ingested because they are very soluble in fat, and because of that trespass the cell membrane very easily, which facilitates their binding to many intracellular lipophilic molecules and therefore interrupting the regular cellular processes. In this they differ from acrylamides and nitrosamines, which do not possess this hyperlipidic character, and therefore they only bind to DNA. However, just as their counterparts, PAHs are also metabolized by cytochrome P450 and other important enzymes that metabolize toxic components, generating highly reactive metabolites like more epoxides and radical cations that will bind to DNA because of their positive charge, also generating the adducts that we mentioned before, which contribute immensely to the expression of proto-oncogenes. Now, polycyclic aromatic hydrocarbons also alter T helper 17 cells and regulatory T cell count. They decrease humoral immunity, they modify membrane of leukocytes such as neutrophils and other white blood cells that are very important for the immune system, and they alter their proper immune role, contributing to a massive immunosuppression that enhances the carcinogenic response of the organism to ingestion of these compounds. So, in the end, all these three carcinogens found in food, especially when consumed through the oral route, they will not only bind to DNA and enhance this DNA damage and also interrupt the DNA repair mechanisms so that they can form this new cancer cells, but they will also form this immunosuppressant environment that will make it even harder for the organism to defend itself against this newly growing cancer.
In this slide, I will be discussing the long-term risks that are associated to ingestion of carcinogens through our food. There are many long-term risks related to the exposure of chemicals in our food and to our health. The main concern when discussing these complications is cancer and the different types of dangerous tumors that can grow in our body after prolonged ingestion and exposure. Carcinogens have the ability to slip past our body's natural filtration system and encourage the mutation of cells in different parts in our body, such as the lungs, stomach, liver, and kidneys, which all have the potential to develop cancer from these chemicals. Other diseases have been linked to exposure, such as autoimmune diseases, which can cause very dangerous changes to the way our body fights off disease and protects itself from biological invaders. Carcinogens have been found to have a detrimental effect on our endocrine system, causing many problems in terms of our hormonal balance and production. Hormonal complications can lead to severe diseases of our autoimmune system, such as lupus, and developmental complications in those who are young. Kidney diseases and rashes can also develop as exposure continues, causing sensitivity of the skin and body to environmental stressors. All of these diseases continue to worsen as exposure to chemicals in our body increases, and the likelihood of developing cancer is the most alarming increase, since we are seeing a large number of people developing cancer, especially here in the United States. There are many other long-term effects due to carcinogens in our food that exist that maybe have even yet to be discovered until we continue to research and observe its effects over time. In terms of future implications, carcinogens within our food supply can go unnoticed for a long time. The longer one is exposed, the more they are at risk of detrimental health problems. Research has shown that 1/3 of all cancers are related to food in some way, and it can be a result of many different types of cancer. Adolescents and young children are considered the most at risk for developing long-lasting effects from carcinogen ingestion. Infants younger than 2 years old are believed to have a 10 times risk factor of ingesting carcinogens, while children aged between 2 to 15 have a 3 times higher risk. This raises the question and concern regarding what future dangers this can hold, since young children in developmental stages of their lives are ingesting all of these chemicals. These stages of life are very critical to the development and long-term health of a young child, and earlier exposure to these children could cause early onset cancer and other diseases not typically seen in young people. As exposure continues, in the future we may see organ function in such as mammary glands, colon, and prostates develop cancer at alarming rates, which all can affect our reproductive health as well. This leads to increasing concerns for newborns and pregnant women and the different complications that develop for both the fetus and the mother after increased exposure to carcinogens in food. For combating this issue, the best way to combat this pressing concern is continued research and identification of the carcinogens present in our food sources and their lasting effects on our body and health. Identifying these harmful chemicals can help us work toward eliminating them from our diets and the risks they are presenting in the long term and short term. We also must continue to encourage our society to move toward cleaner environmental practices and the elimination of possible pollution and chemical introduction into both food and water sources and within the air we breathe as well. Introduction of cleaner energy and industrial activities can help limit and lead to the elimination of carcinogens entering our body through this manner. The environment surrounding us requires commitment to creating policies encouraging cleaner practices and removal of carcinogens from our diet to ensure a healthier lifestyle for all, preventing this additional risk of cancer. Ensuring and promoting a better quality of life is extremely important, and we all have to use our voice to the government to continue and improve the treatment and policies surrounding both cancer research and how to stop carcinogens from entering our food and body. To discuss, understanding the potential issues caused by carcinogens in our food is vital to our survival and ensuring a healthy and long life for each individual as much as possible. Prolonged and extensive exposure to these carcinogens not only affects us as individuals, but also carries an extreme risk down to our children and the generations who come after us. Our society and government as a whole must continue to strive to do our best to implement and encourage change in our policies to promote cleaner environmental practices and removal of dangerous carcinogens from our foods. If action is not taken soon and quickly, we could soon see a dangerous increase in cancer rates in young people and a poor quality of life for those affected by such exposure to carcinogens. Increased cancer risks not only place pressure on the individual, but the medical field and economy as well, to ensure that proper and affordable treatment is available to those who continue to become affected by such exposure. It may take a lot of work and a long time to truly implement these types of permanent changes within our society, but there is something that needs to be done in order to ensure people do not develop cancer and other similar diseases that could lead to their death as a result of this manner, possibly at an even younger age than we have ever seen before. Practices and protections must change to avoid and limit carcinogen ingestion from our food.